Adults have approximately 5 liters or a gallon and a quarter of blood in our bodies. We can only tolerate no more than 4 grams or 1 teaspoon of sugar in our bloodstream at a time. Three problematic issues arise with chronically elevated levels of glucose and insulin from continually eating highly refined carbohydrates. First problem: ultra processed foods contain many additives and chemical substances that our body does not recognize and does not know how to metabolize. Instead, these chemical substances destroy the cell membranes, leaving them dysfunctional, causing system wide irritation and inflammation. Second problem with high ultra processed and refined carbohydrate intake: more insulin remains in the bloodstream, reaching toxic levels, causing more irritation and inflammation, and preventing channels in other cells from allowing insulin to function and get glucose inside. As more glucose channels and insulin receptors are destroyed with excess blood insulin levels, the insulin receptors start to downregulate, resulting in a decreased response to insulin, leaving higher levels of glucose unattended in the bloodstream. This is, in essence, what insulin resistance is. Third problem with high refined carbohydrate intake: the high levels of glucose that remain in the bloodstream when insulin receptors have downregulated, a phenomenon known as glycation occurs. The sugar in the bloodstream starts to coat proteins, which makes proteins sticky. Proteins are responsible for the structure and many functions in our body. And the glycation that occurs when glucose sticks to proteins makes our tissues and organs, including our brain, more dysfunctional. We test glycation in our body by measuring our hemoglobin A1C, with optimal levels of glycation to our hemoglobin being 5% or less, and why we are concerned when levels reach 7.0% or 10%, which is a relative increase rate in glycation of 30% and 50% respectively. If you can imagine this sticky glycation happening to proteins in our brain, in our heart, and our reproductive organs, which reduces cell communication, causing major organ and tissue dysfunction.